Howdy, I'm Steve Saltwick, and welcome to my seminar, Your Brain, A Personal Tour. What are we looking at? Well, it's called the Edward Smith Surgical Papyrus. It's a papyrus from about 1700 BC, or in other words, around 4,000 years ago, that describes the ailment and prognosis of a patient by an Egyptian surgeon. It is the first recorded use of the word for brain, and it means that we've studied the topic of the brain for many thousands of years. But hold on. Why should we study the brain at all? Isn't that the fundamental question? Well, and the fundamental answer is to advance science. The brain is the most complex organ in our body. And as we've learned, we've studied it for a very long time. And we've learned a lot. And let me give you some examples. When we advance science in our knowledge of the brain, we increase our ability to recover from trauma. Shown here on the right is a tweet from an ALS patient who previously could not communicate at all. But with a Neuralink implant and training, he is now able to communicate, although some would argue tweeting is not communicating. This capability represents a betterment of his quality of life that broaches on the miraculous. It is a wonderful thing that such a device is now designated by the FDA as a breakthrough device. Such a designation will expand the reach of this breakthrough and relieve the sufferings of many more patients. Now, I like to be a bit playful in my seminars, and I will share that we study the brain not only to advance science, but to give people snappy patter for cocktail parties or dinner parties. For example, holding a cold drink in your hand will bias your opinion of someone else. Nuns do something that can help prevent Alzheimer's disease. 6-month-old babies make moral decisions. And there are single cells in your brain that represent Jennifer Aniston. And not to be sexist, could be Ryan Gosling or Homer Simpson. How can this be? Well, we study the brains of humans and of other animals, especially the rat. You're going to hear a lot about rats in this seminar. Why? Because humans do think like rats. By that, I mean the general mechanism of thinking are the same between rats and us. Now, we do think about different things. I doubt rats think about Jennifer Aniston. But the reason I will talk about rats is because the study of their brain will eliminate so much of our brain. And that's why we're in the seminar, right? Now, I know many people like to take notes, and please feel free to do so in this seminar. It turns out that handwritten notes are one of the best ways to remember something, and it turns out also that typing the notes is not such a good way. However, there's some significant learning aids that I want you to be aware of. First, all seminars are on YouTube. A link to the YouTube playlist is in the campsite information on this course. So if you miss an episode, don't fear, you can catch up on YouTube. There's also a reading list on Campsite. Each seminar, I will present 1 or 2 books to be explored for anyone who wants to go deeper in the topic. And finally, there'll be a link and a QR code to an AI-generated podcast, typically about 15 minutes long, that summarizes the seminar. And I'll present that at the end. If the slides are overwhelming, or you just want to refresh your memory in the car someday, then the podcast is a great way to go. The voices are outstanding.
Hey everyone, and welcome to another deep dive into, the most complex structure we know of.
And if you're wondering how I got into the brain business, well, I became first interested in this marvelous organ in the biopsychology area of the psychology department of UT Austin. Here's a picture of us in 1975 in front of Macy's Hall. I was privileged to work in the lab of Dr. Michael Gabriel, and we were fortunate in the pursuit of some exciting brain research that was published in the journal Science. I left UT in the late 1970s and took a tour through the high-tech industry. It was great fun, but even more fun was returning to UT after my retirement and reestablishing contact with several of my major professors on my dissertation committee. Dr. Michael Gabriel, Dr. Michael Damian, and Dr. Dennis McFadden gave me thorough and constructive reviews of the early drafts of this seminar. This seminar would not have been possible without their help and encouragement. And if you're wondering, where's Steve Salwick in this picture? Well, here I am with a little more hair and attitude and a lot less weight than the one in front of you today. So let's now turn to our first topic, perception and movement. I try to summarize the key thought of each seminar segment with a subtitle. And for this seminar segment, it's what you see is not what you get, nor how you act. And I also want to draw your attention to the small print at the bottom. I am not a medical doctor. Do not take this seminar as medical device. It is designed for educational purposes only. Now, to get a better handle on the meaning of the statement that we have as a subtitle, we need to go back to that papyrus. 4,000 years ago, the thought was the brain was pretty much an undifferentiated mass, kind of like the liver. But now we know there's functional areas, and we can use that knowledge to repair lives. For example, the life of violinist Roger Frisch. Roger had developed a tremor. Not too bad for you or me, but for a professional violinist, devastating. So since we now know about key functional areas of the brain, science could help with a specific procedure.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
Now, there are no pain receptors in the brain, so Roger was under no distress when this operation was being done, even though it looks really scary. Now, to see the impact on Roger's life, let's take a look at this film clip.
You can see the difference. With the click of a controller, Roger can turn the electrodes on and off.
And that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin.
Now, your brain is part of your nervous system, a vast network of nerves that course throughout your body, your muscles, your organs, and so forth. We'll just not have time to study the full nervous system. However, there is one nerve outside your brain that we will study, the vagus nerve. This nerve connects your internal organs, especially your gut, with your brain. Much recent research is centered around how this nerve is implicated in Alzheimer's disease, Parkinson's disease, and others. It's just too important to leave out, and we're going to get to it in Seminar 6. Your brain is made up of building blocks of cells. The most common in the workhorse is called the neuron. There's over 85 billion neurons in the average human brain, more than there are stars of the Milky Way. The neurons talk to one another via connections called synapses. A neuron fires an electrical signal, shown here in blue, down a long tail. That electrical signal gets to the end and causes a chemical to be released. That chemical affects the next neuron in the chain. It may make it fire, in which case we say it excites the next cell, or it may make the cell less likely to fire, in which case we say it inhibits the cell. We can listen to the activity of brain neurons by placing a listening device, called an electrode, into the part of the brain we wish to study. When a neuron is firing, we hear clicks on the output of the recording device, like this. Since neurons use electricity to send signals, we can influence those signals with magnetism for the release of suffering. Here's an example. Using magnetic stimulation from a device pictured on the right, we can alleviate the symptoms of depression in many, many sufferers. I have a personal friend whose son's depression was treatment-resistant to drugs. He tried this kind of stimulation and is symptom-free. I do want to take a closer look at how two neurons talk to each other. The electrical signal shown here as a traveling series of white dots is converted to a chemical signal, and that chemical signal is released across a small gap between the cells. If that chemical signal is sufficiently strong, then a new electrical signal is generated in the neuron across the gap, and the signal continues. If not, the signal stops. I want to show you a high-resolution photograph of a real brain cell. We'll look at a lot of simple pictures in this seminar, but I want you to keep in the back of your mind that the connections are far more complex than we show in the schematic diagrams. Now, let's look at our first section of the seminar, sensing and perceiving. Remember that the brain is fully encased in bone, the skull. So how does your brain get information about the world outside your skull? This is the realm of the senses. While this may seem simple, it's a very complex problem, and we'll see that your brain may sense many things that you are unaware of at the conscious level. What you perceive consciously may be very different than what you sense. At some point in life, we typically reviewed the senses as shown here on the slide. These senses are wonderful, and they bring in a rich set of information to the brain. Specialized nerve endings transform light, sound, chemicals in the air into vision, hearing, and smell. Each sense has a prime sensory area in the brain. Noble as these sensors are, we'll not have time to investigate each one in detail. There's simply not enough time. But I want to look at a few that you may not be that familiar with. For example, the somatic senses. And these senses have surprising impact on the brain. Let me give you some examples. We have temperature and strain gauges in our fingertips. The sensations from these senses can actually bias our perceptions. If we hold a cold drink in our hand, we will judge someone we're talking to as cold in temperament. And yes, the same for a hot cup of coffee. If you want to make friends with someone, buy them a coffee or hot tea. If you read someone's resume on a clipboard, you'll judge them as more serious than if the resume is on a light piece of paper. Another hidden somatic sense is the sense of the body in the state of muscles, called proprioception. Each muscle is richly innervated with something called muscle spindles, which convey information on the state of the muscle. These spindles, coupled with receptors in our joints and some in our skin, give the brain extensive information on the state of the body in terms of posture and movement, particularly the four limbs and the head. It turns out this is a major sense for the brain, although we're unconscious of it most of the time. It may be a main source of information used by the brain to construct the concept of self, and when processing this self goes awry, people may conclude evil agents are moving their limbs, etc. More on that later. Proprioception can influence our perception as well. Using a unique experiment, scientists can show that smiling is mimicked by holding a pencil in your teeth can reduce your negative bias against someone. So smile. You will feel more positive. But when all is said and done, humans are visual animals. Your eyes are populated with a rich set of sensors that turn light into nerve activity. That activity is transmitted fairly directly to the back of the brain. Here we see a cut through the brain that describes this pathway. The cut is like you took a chef's knife and chopped off the top of the skull just above the eyes. The area in the back of the brain is called the primary visual cortex, and it is here that visual information is processed at a low or elemental level. The primary visual cortex dominates the back of the brain so much that this whole area of the brain is called the occipital lobe. All parts of the eye are not equal. The center of the eye is very sensitive, and it's the only part of the eye that can sense color. Your brain moves your eyes over the visual field to fix the center of the eyes on interesting parts. It's probably easier to show, so let's see an example. Using computers and mirrors, we can depict where the brain is fixing the center of the eye. That's the red dot in this movie. Let's take a simple scene and see where the eye concentrates. So where did the eye focus? Faces. Did the eye ever look at the whole scene? No. For example, it never looked at the clothes in the closet, etc. This aspect of visual processing will have significant effects on how we make decisions and remember events. But let's not get ahead of ourselves. The net of this slide is that the brain gets selected information of a visual scene. Our brain has to construct the rest. Here's an interesting example of the filling in process. Look at this graphic. See the small dot at the intersection of the lines? There only seems to be one or a few of the dots. But then look at a different area of the graphic. Is there a dot there? And look at a different area. Is there a dot there? What's happening? Well, as the quote shows, your brain is filling in what it thinks is going on. It's guessing. So the eyes are not lying, but our perception is not getting the full picture. And that leads us to perception and consciousness. When do we really become conscious of something in our visual world? Well, let's take some more examples. In this video, we're going to see some flat lines become three-dimensional figures. The lines do not change. Our perception of the line changes as we build our visual world. Let's take a look. As we construct our visual world, we're constructing objects. And those objects have three-dimensional form as best as we can see. We're perceiving almost solid objects, while in reality, our eyes are getting just flat lines on a piece of paper. And it turns out our brains build objects like the 3D shapes in the previous slide, and those objects can be represented in our brain by single neurons. Some of those neurons might be Jennifer Aniston, or Ryan Gosling, or Tom Cruise, or Homer Simpson. Now, we're not born that way. We learn them. From a very early age, our brains are learning to build ever more complex objects. On this slide, a 6-week-old baby will judge these two figures equal, since this line and this line are judged equal. By 14 weeks, these two figures are judged the same, since the angles are the same. Angles are a more complex object. Many scientists believe that when your brain constructs objects from sensory data, you consciously perceive that object. Let's take another example. When you look at this slide, most people report a triangle outlined in blue under a triangle of pure white, which is also over a set of 3 circles. Anyone disagree? Notice what happened. Your brain assembled the basic lines and forms and put them into objects, triangles, circles, in a spatial arrangement. And most importantly, your brain decided on the spatial organization of that physical world. What was over what? This is not trivial, and many think that this is exactly when consciousness of the scene occurs. Your brain has assigned an object, your brain has determined the spatial arrangements, and very important, if you have to reach for the triangle under the white one. Once your brain constructs the object, it can be very convincing, especially if that object is perceived to be part of your body. Let's take a look at an example.
I have an experiment that we're going to do today. Is I'm going to train your brain to believe that this hand, this arm, is your arm. You see we've got this divider here? So right now you can kind of see your arm. You see the arm there? And I'm going to cover over part of your arm. Now I'm doing that because your eyes talk to your brain. And your eyes tell you this is what's happening. So now, can you physically see your arm, your right arm here?
No.
Great. So I'm going to take these two classroom rulers. So we're going to train the sensations. You feel the rulers touching your hands because it's now becoming one with your mind. That this right hand is your right hand, right? You are believing that as I drag the ruler on your right hand there,
It
that
totally
it is
feels.
in fact in your brain your
yeah.
right hand.
It totally feels like my hand. That's crazy.
Feel the sensations
Yeah.
line up with each finger.
What? Whoa, yo, this is crazy.
Yeah. So now if I just do like a little tap, right?
Yeah.
Feel the knuckles. Feel each digit.
Oh, that's
Okay, this is where.
crazy.
Yeah. Now I'm gonna do this one more time to train your brain.
Okay.
Right. So you feel your thumb
Oh, okay, yeah.
and your index finger, middle finger,
This is nuts.
your ring finger,
Oh my goodness.
and your pinky finger. And now you're gonna feel it when I tap it. You feel that?
Yeah.
And that. And
Whoa.
that. I'm gonna let you in on a little secret.
Yeah.
I have not been using the other ruler.
What?
You feel that?
Yeah.
I am not using the ruler.
Whoa. What do you? I mean, it's crazy. It's not. Oh. Whoa.
You
It
felt
even
that,
like.
right?
Oh no.
Now, check this
Oh.
out. We're gonna do a little test. Now, again, this is all in your mind. I'm retraining your brain to feel sensations.
Okay.
But your brain is thinking that it's feeling it.
Okay.
So even if it gets a little, what would be considered painful, I will not be harming you because of course I'm not going to be actually hitting your right arm. But your brain really believes that this is in fact your right hand.
Oh,
right?
this is making me nervous.
All right, here
Okay.
we go. I'll start lightly. Feel that on the thumb.
Whoa. Yeah. Oh. Whoa.
Ring finger,
I'm totally feeling it in this hand. It feels.
pinky,
It just. Oh. Wow, this is so
back of
strange.
the hand. Do you feel that?
Yeah, I want to move my hand, but it feels like I can't. What's that for?
Now again, I would never on purpose hurt you, right?
Okay.
And I would never take a hammer and bash your fingers to where they would hurt, right?
I'd hope not.
But again, your brain is going to tell you that you're going to feel this when in fact I'm not actually going to be causing any damage to your hand. Right?
Okay.
And we'll start lightly again. Okay?
Okay.
You ready?
Yeah.
Here we go. Very lightly. Here we go. There.
Oh, it's so weird. Oh.
And oof.
Oh.
Oh, the infamous thumb when you're hammering. Oof. Missed the nail.
Oh, you know, it feels like an electric shock. It's so strange.
Doesn't it?
Yeah. Oh, even when I touch myself.
Yeah.
Oh.
Yeah. Watch this. Watch this. One last electric. Watch this. You ready for this one?
Okay.
Guess which finger I'm going to hit.
Okay.
Guess which finger. See if you can tell me which one it is.
Middle.
Now again, I did not actually hurt you, did you? You didn't feel
No.
actual pain in your real hand. But For an instant, your brain told you that you were thinking that you felt a hammer hitting your hand.
My brain thought that this was my hand.
Now, one convention we'll use during the seminar is that each major section will have a section takeaway slide. And we're going to use Homer Simpson's view to give us the essential, view or key message for each of these major sections. So what does Homer say about this particular section? Homer says you sense stuff and you perceive objects which you predict. So this gets back to our, subhead. What you see is not what you get. Now, let's transition to the second aspect of the seminar, and that is movement. The question I want to begin with is, are there movement objects like what we see in the perceptual system? The short answer is yes, but I think it's better to think of them as a vocabulary, and we'll see why shortly. There's some evidence for movement objects, and it comes from, experiments. For example, a famous experiment back in 1977. The subjects were asked to write a famous palindrome, which means a sentence that's symmetrical about its middle, with a variety of methods. Even though vastly different muscles and limbs were being used, it's astonishing that the palindrome is clear no matter what the mechanism is of writing. This was taken as consistent that an object of the sentence was held in the brain and then transformed to specific motor movements by other parts of the brain. Perhaps the most elegant experiment that conclusively shows there are objects in the brain for movement was published in 2007. Here, monkeys reached and grabbed for a food item using one of two sets of pliers. In one case, the normal pliers, the monkeys had to relax their grip to open the pliers and to squeeze the pliers to lock onto the food and bring it to their mouth. In the other case, the reverse pliers, the monkeys had to do the opposite. In other words, squeeze to open the pliers and then relax their grip to lock onto the food. Consider, the goal of the movement in both cases was the same, lock onto the food. However, the exact opposite movements were required in the two cases. Relax and then squeeze versus squeeze and then relax. Let's see what happened. Here we're going to see two video recordings of the monkey. In the top left hand, we're seeing the monkey using the normal pliers grab the food. The food is yellow, and we're tracing what the monkey is looking at with red lines coming out of his eyes. This is not Godzilla monkey. This is a computer animation of what the monkey is looking at. On the bottom left is the monkey using the reverse set of pliers. Same kind of setup. We can see what the monkey is looking at and watch him as he grabs the food. Now, what did we see in the brain that was happening when the monkeys did this movement? On the top, we see two different brain cells. One is called Unit 210 and the other is called Unit 199. We map the behavior of this neuron, or each of these neurons, in two different ways. In the graph that you see here, there's a tick mark at the top for every time that neuron fired. So in other words, every time the brain neuron got excited and fired, there's a small vertical dash at the top of this diagram. And we'll show those vertical dashes both for Unit 210 and for Unit 199. Immediately beneath those dashes is a histogram that sums those dashes over time. Each row of the dashes is a separate movement by the monkey. So what we can see is that there's a peak of those neurons' activities, and that represents precisely when they are grabbing the food, in this instance with the normal pliers. Now, what did those same neurons do when they had to use the reverse pliers to grab the food? And remember, the actual muscle movements now were completely different. In fact, they were the opposite of what the monkey was doing in terms of movements in the top figure. Well, what we see is these neurons behaved exactly the same way. I would argue that these neurons represent the object of grab the food. It's independent of muscle movement. It represents the actual movement object itself. That movement object is then translated to a variety of muscle movements by appropriate other systems of the brain. This slide summarizes research from several studies which stimulated parts of the motor cortex and the frontal lobes of the monkey. You can see from the left side of the slide that there were several discrete areas which, when stimulated, led to various coordinated movements: reaching to grasp, leaping, moving, hand to mouth, etc. I assert that these types of movements are clear parts of a vocabulary or movement objects which are represented in the monkey brain. How do words, in other words, high-order movements, get transformed into a specific set of actual muscle movements? Well, as shown on the right, it's rather complicated. The right side of this slide, in fact, is very complex. We're not going to have time to go into this in detail. Research in the fundamentals of movement are proceeding at a rapid pace. With the advent of computer analysis of real-time behavior, we're forming a language of movement in rat and mouse brains. Human brains are close behind. Remember the tweet? This research can have an almost magical impact. And here is that example again of Kevin Petit. Kevin was a quadriplegic. He is a quadriplegic. Until lately, he could not walk. However, thanks to advances in brain science and manufacturing, he was able to carry an Olympic torch in 2024. I want to close this section with a couple of points. The first point is, what is the difference between doing a movement and thinking about doing a movement? Well, this slide shows you one example. The top part is the active parts of your brain when you do a sequential finger movement. For example, like strumming your fingers. The bottom part of the figure is when you think about doing the finger strumming without moving your fingers. My main point here is that when you imagine moving your body, it activates part of the same brain area that participate in the actual movement. This is an important point, and we'll return to it when we discuss memory in Seminar 3. And that brings us to our Homer slide. What's the section takeaway of the movement? Is that you plan with movement objects, and then you move muscle stuff. The exact opposite of what we do with our perceptual and sensory systems. It's rather elegant, is it not? Now, before we set up the next seminar, I want to talk about one aspect of your motor system that I want to highlight, and that is the motor system can know things that you don't. What do I mean by that? Well, let's take a look. This figure is called the Ebbinghaus illusion. I hope everyone sees a bigger orange circle on the right compared to the left. Is that true? I think everyone knows that the circles in the middle are the same size. You've probably seen this illusion before, but your visual system is tricked by the presence of the outer circles. Now suppose I model these same circles with wooden blocks that you can pick up, and I ask you to reach out and grab the circles, one after the other. You would open your thumb and fingers exactly the same for both of the orange circles. Your motor system, as it's planning the reach and grab, uses different systems in the brain to calculate how much to open your fingers. These systems are not the same as those used by the visual system. So part of your brain knows the truth, and part of it is confused. It depends whether you just look or you look and reach. This concludes Seminar 1. In Seminar 2, we'll look at the decision to act. and I'll motivate that seminar with a famous quote from Amelia Earhart. And Amelia said, "The most difficult thing is the decision to act. The rest is mere tenacity." We'll look at how that statement from Amelia is embodied in your brain and the 4 different ways your body decides to act. This is Steve Saltwick. I hope you've enjoyed Seminar 1.